Yeah, it's how I feel. Okay, everybody out there, very good afternoon to all of you. I hope you can all hear me. If anyone cannot hear me, please let me know. Okay, so just with the chat function, let's see, anyone there? Hopefully, good. So. And let's get started. All good, yes, brilliant. Love that. Thank you very much for the feedback. Good. Okay, most wonderful, good. ICT time, because that would have been your in-class test. But I mean, to be perfectly honest, it is pretty stupid to sort of arrange for a test on a topic when you had only, how many? 4 lectures or something like that on a topic, because that is supposed to be on the metabolism. So I hope you don't mind that we don't do an in-class test and instead replace that with some of the multiple choice stuff. Okay, I don't hear a resounding objection to that suggestion, so no in-class test for that. But may I suggest to you, very sort of quietly, gently, carefully, that when you do the relevant multiple choice test for this metabolism, you really take note of it, of the questions, of the answers, because I think this is a great learning opportunity. And just in case you are not aware, the exam for this module, which counts for 50%, I think, consists of a section A, which is 30 multiple choice questions on metabolism. Who would have thought that? And section B is a problem question about enzymes. So my gut feeling tells me somehow that the multiple choice quiz might be somewhat related to the multiple choice question in section A in the exam. It's just a, sort of a suggestion. So please do the quizzes. They're open until I can't remember when. I think it's probably 22nd of March. I don't know. People, I'm very grateful to some people who contacted me saying that there was a problem with the BI 308 quizzes, at least 2 of them. I have not changed anything for these quizzes, so my suspicion is that Mr. Google has fluffed it a little bit, but I think I have sorted out the problem, but please let me know if there are any issues. Right, now what are we going to do today? Today I want to discuss with you some, if you like, absolutely crucial things when it comes to metabolism. And let's imagine you are, let's say, a skeletal muscle cell. Now, last week we've discussed glycolysis, and we discussed how a muscle cell actually can utilize glucose, which it usually derives either from the blood or from glycogen stores. And it utilizes this glucose in order to gain energy, to produce ATP. Although glycolysis effectively is not terribly good at producing lots of ATP, because we only get 2 moles of ATP per molecule of or mole of glucose and 2 NADH. So it's not terribly effective, but we will next week, we will see how we can vavavoom the ATP output. But one of the problems is the cell has to make a decision whether it should use the glucose either to get ATP from it, or whether it should store the glucose in the form of glycogen, sort of tuck it away for a rainy day. And I told you that skeletal muscles store huge amounts of glycogen, about 400 grams of glycogen. The liver has about 100 grams of glycogen. So the typical place to find glycogen as an energy resource is skeletal muscle. Now the muscle has to make this decision. Am I going to eat it, or do I put it in the fridge and store it for later? And that brings us to the topic of regulation. There are lots of different ways how reactions, how metabolism can be regulated. What I want to show you are some very typical types of regulation, which we will come across in the next couple of sessions. Now, we have already seen that one type of regulation is that you can express isoenzymes, enzymes that catalyze the same reaction but have a different polypeptide chain, that you can express them in different cell types. And I told you hexokinase 1, 2, 3, these are the starting enzymes for glycolysis, are usually expressed more or less wherever, But hexokinase 4, which is commonly known as glucokinase, is only expressed in the liver and a few rare other tissues. How does that happen? Well, very simple, because every cell, whatever cell it is, every cell has its own unique profile of genes that it expresses. And that is achieved by promoters that are tissue or cell specific. We can also express different isoenzymes in different organelles. So, for example, we might find that one of the key regulatory enzymes in the opposite of glycolysis, in glyconeogenesis, is expressed only in liver, some kidney cells, some intestine cells, and it is only present, this particular enzyme, in the endoplasmic reticulum or other enzymes in the mitochondria. So that's one way. Another way is that enzymes can be regulated, and we have already heard about regulation of enzymes in terms of negative, what I call negative effectors. And a negative effector just simply is the same as an inhibitor. And we have done inhibitors quite extensively. We discussed the competitive inhibitor, where the inhibitor only interacts with the free enzyme, either by binding to the active site or a different site on the enzyme. But the point is that it interacts only with the free enzyme. We have discussed uncompetitive inhibitors, where the inhibitor only interacts with the enzyme substrate complex. And we have discussed mixed inhibitors, where the inhibitor can interact either with the free enzyme and also with the enzyme substrate complex. So these are negative effectors, but very often we also find the opposite. We find positive effectors. And what these positive effectors actually do is they bind to an enzyme and they would then activate it. So they are activators of the enzyme. How do they do that? Well, usually the enzyme has a sort of a binding site or a regulatory site where the activator binds. Binding of the activator changes the structure of the enzyme somehow and makes it, for example, more interested in the substrate. Or it induces a change in the enzyme so that it can deal with the substrate quicker. Or it can do something to the enzyme so that it releases the products quicker. So these activators can do very different things. And what we find, and I will give you an example, is that that is our little enzyme. That's the catalytic subunit. Where all the action happens, and you see the little mouse, that's the active site. And you can imagine that you could have a little foot there, a little shoe, and that might be the regulatory subunit. And this regulatory subunit can be the same polypeptide chain, or it can be a separate polypeptide that is just simply attached to that. Sometimes, if they are separate, then with very gentle denaturation methods, you can separate them. And what you end up with is only the catalytic subunit, which of course in this case would no longer be in any way regulated. But in order to be able to turn this thing on or off or whatever, you would have to have them both together. That is called a holoenzyme. Holo means all together. We also distinguish. So this regulatory subunit can do things, can, say, induce the enzyme to work faster, to do different things, to release the products quicker, etc., etc. We distinguish between hetero, Homo, Tropic effectors. And the difference is not terribly difficult. A heterotropic effector is a metabolite or a compound that is not the same as the substrate. And I will give you an example for that in a minute. The important thing is it is not the same as the substrate. A homotropic effector, therefore, would be, it is the same as the substrate. So in the case of, for example, phosphofructokinase 1, PFK1, where we know it catalyzes the reaction of fructose 6-phosphate plus ATP, gives fructose 1,6-bisphosphate plus ADP. ATP, and that is one of the key reactions in glycolysis. This is one of the, what is called the rate-limiting reactions. That's one of the slow reactions in glycolysis. One of the substrates is ATP. That's the substrate. And at the same time, ATP is also a negative effector sort of an inhibitor. And in this case, so it's a homotropic effector. And if you think about it, in a way it makes sense, because glycolysis is meant to produce ATP. But if we already have a lot of ATP there, there's no point in really going down the route of producing more ATP. So therefore, it makes sense that this ATP is also used as a sort of a mechanism to say, hey, PFK, wait a moment. We already have enough ATP, so don't work yourself into the grounds, right? So in this case, this is a homotropic negative effector. What does a positive homotropic effector do? We find positive homotropic effectors very often when we have multimeric enzymes. So for example, a tetrameric enzyme, which is made up of, let's say, 4 subunits like that. And what a homotropic positive effector does, when we use here the effector concentration, and here the activity, for a homotropic effector, the first substrate binds, but the binding is not terribly good. The reason for that is because most or all of the substrate, all of the subunits of the enzyme are in what is usually called the R state. No, sorry, not the R state. It's in the T state. T stands for tense, and you know that from yourself. If you are tense, you don't want to bind anything, right? So at very low substrate concentrations, very low concentrations of the effector, the enzyme is not terribly active. So it's probably going like here. However, if you add more and more effector, the T state then can change into an R state. This is the far more relaxed state. Yes, it's good. Life is good. Yeah. Hey, bring on the substrates. Yeah. That's not a big problem. Yeah. Come on. Good stuff. What you see is that as soon as you reach a sort of a critical threshold, the activity of the enzyme goes up until it reaches then the full saturation. And there are different models how you convert from T to R and R to T. Some enzymes show that when you have just one substrate, if it manages to bind to a T state, it will convert the whole thing into an R state. So each one of these 4 subunits will immediately switch to an R state then and bind easier. Other enzymes don't do that. They do it successively, but every time you add more substrate, it gets easier to change from T to R. I think you have done something like that with T and R states in one of the chemical biology assessments. Did you? Where something changes? If not, don't worry. So this is what homotropic effector does. And actually, if you think about it, this is really a nice, if you like, by the way, this is called a sigmoidal shape, or S shape of that curve. And what it does is, it is also very often quite useful as a sensor, because the cell can sense how much of the effector, how much of the substrate is actually present. So at a certain threshold, the cell says, before we reach that threshold, no, we are not going to do anything, right? In this range here, the response of the cell is directly proportional to the amount of the substrate that's there. And then in this range here, basically the enzyme is always switched on. But this range here, let's do that in a different color. This range here is really the sensor range, where the cell can actually measure how much of the substrate do I have there, and it can adjust its basically its activity of the enzyme accordingly. That makes sense? Fantastic. Okay. Now let's have a look at PFK. As I said, phosphofructokinase 1 is one of the key enzymes in glycolysis. It will determine whether glycolysis actually happens or whether it doesn't happen. And as I told you, it's quite important that the cell makes this decision, because if energy is there, if the cell has energy, there is no need to use up precious glucose. Instead of, we put the glucose into storage. And how is that done actually? Now, PFK1, phosphofructokinase 1, in the cell is usually pretty much inactive. It doesn't do a lot. It sort of plots along. Yeah, there is some glucose, there is some fructose 6-phosphate hanging around. Yeah, hey, who cares? I'd rather, watch Netflix and chill and all these things. I don't give a damn. No, not today. So I'm not in the mood for doing any, conversion to fructose 1,6-bisphosphate. I can't be bothered. How do we actively get this thing active? PFK1 has a positive effector. This one would be, in this case, a heterotropic effector. And this effector is, I better write that out. Fructose 2,6-bisphosphate. Or F26BP. Fructose 2,6-bisphosphate binds to phosphofructokinase 1 and activates it. It basically gives it a kick in the backside and says, mate, do something. And only when fructose 2,6BP is present in the cell, the reaction, it can activate PFK1 and glycolysis will happen at a reasonable speed. Only then. So where do we get this activator from? Well, we start with the same substrate that PFK1 uses, namely fructose 6-phosphate. That's the same thing that PFK1 uses, and we convert that with the use of ATP. But this time, we do not attach the phosphate to the one group, to the one residue in fructose. We attach it to the 2 group. Please enlarge the screen. Okay, good. In this case, I can enlarge the screen, but then you won't be able to see the chats, and I won't see them as well, but I'm happy to do that. Better? Good. So, we transfer a phosphate residue onto F6P and get F26 bisphosphate. This is no longer a substrate for PFK1. It's not a substrate. It's not a substrate for glycolysis. Glycolysis can't do anything with F26BP, but it is the activator. And the enzyme that actually catalyzes this reaction is called phosphofructokinase 2. It's a totally different enzyme to PFK1. It's easy to remember because PFK2 has a 2 here, and it produces fructose 2,6-bisphosphate. So we have a phosphorylation reaction, and this product now can activate PFK1. That's what its job is. And then we also want to turn off the system if energy is abundant. Then we don't need PFK1 anymore. We don't want the glycolysis to happen, so we convert F26 bisphosphate back into fructose 6-phosphate. And we do that by just simply getting rid of an inorganic phosphate. The enzyme that does that is a phosphatase. Here we have a kinase. So PFK2 is a kinase. It transfers a phosphate residue onto F6 phosphate. And a phosphatase does exactly the opposite. It removes the phosphate and it turns off F26 bisphosphate and converts it into F6 phosphate. And what people discovered is that this is a very, very common theme in cells. So for example, we have over 200 genes that encode kinases and about the same number of genes that encode phosphatases. And when people saw that, when researchers saw this for the first time, they thought, that is mad. That's total madness. It's a little bit like, how shall I describe that? My wife earns all the money, and I spend it all. In, out, in, out. It's basically, if you look at it, it's a fantastic way of destroying ATP. What it basically does is it converts ATP into ADP plus phosphate, the reaction, just simply by cycling. And therefore, people originally gave it the name of a futile cycle, because it's not doing anything. It's just rubbish. It destroys precious ATP. And then people thought, but there must be a reason for that. So they did some computer modeling and tried to figure out what happens. And they discovered that when both kinase and the phosphatase work close to Vmax. And we know that when an enzyme reaction is close to Vmax, we will never get total Vmax, but we can be close to Vmax, then we are working at a zero-order reaction. If we are close to Vmax and have a zero-order reaction, what this system actually does is the following. So here we plot activity, and on the x-axis we plot the ratio of kinase over phosphatase. The two antagonistic things, kinase over phosphatase. Now, when they're equally strong, it's difficult to have the system well defined. But imagine in our system here, if the kinase is stronger than the phosphatase. So if the kinase is stronger than the phosphatase, which of the compounds will we get? Which of the compounds do we get if the kinase is stronger than the phosphatase? Which compound do we get? What is the compound that we get? Loud? What is the compound that we produce if the kinase is stronger?
Fructose 2,6-bisphosphate.
Indeed, absolutely right. We will get that guy. And actually, it turns out we don't get anything of the F6P. Because as soon as we produce a little bit of F6P in the reverse reaction, it is immediately converted back to the F26P. So if kinase over phosphatase is larger than 1, we have basically no F6 phosphatase, and we have everything as F26 bisphosphate. On the other hand, if the phosphatase is stronger, so kinase divided by phosphatase is smaller than 1, what we see is that we go that way if the phosphatase is stronger. So therefore, everything will be in the F6P state. And nothing will be in the F26P phosphate state. What you have here is the typical example of a switch. You change the state like a switch. It's an all or nothing. It's either fructose 6-phosphate or fructose 2,6-bisphosphate. There's nothing in between. It's all or nothing. And that is an incredibly clever way of dealing with that. This is also called, because we are at zero order, this is called zero order. And because the system is incredibly sensitive, you just need a tiny little change in the ratio of kinase to phosphatase to get this switch done. It's called zero order ultra sensitivity. Does that make sense? Now, in the case of PFK2, we have a nice little added level of complexity. Because PFK2 can actually do exactly the same two things. It has kinase activity, and the same molecule has also phosphatase activity. That is why this enzyme is very often referred to as bifunctional enzyme. PFK2 is a wonderful example of a bifunctional enzyme because it has kinase activity and it has phosphatase activity. But then how on earth can we get this switch system to work? Actually, and we need the switch system to turn off or turn on glycolysis. Actually, what we do is we use yet another switch system. We have phosphofructokinase 2, and if it is in this state, the kinase activity is active, and the phosphatase activity is inactive. But if we use another kinase, in this case, the kinase is called protein kinase A. It uses, again, ATP. It actually adds a phosphate group to the enzyme. And now it changes the behavior. Phosphate group is negatively charged. It induces a conformational change in the enzyme. It makes the kinase inactive and the phosphatase active. So just simply by adding a phosphate, we reverse the properties of PFK2. And of course, we have a protein kinase, we have a kinase. We also have another enzyme, which is called phosphoprotein phosphatase. Which removes the phosphate. And again, you have here, what you see is another switch system. You shuttle between the phosphorylated state and the dephosphorylated state. You destroy ATP, but what you get is another zero-order ultrasensitivity. You have a switch with which you turn on the switch, if you like. Now, what is the story with protein kinase A? Now, that's an interesting one, because protein kinase A usually, which is abbreviated as pKa, pKa is an enzyme that is made up of a catalytic subunit and a regulatory subunit. Let's call them C and R. Protein kinase A usually is inactive or not very active. And the reason is this regular subunit is a negative effector. So this negative effector inhibits pKa. However, under conditions where the cell needs to switch from using the glucose, or the body needs to switch from using glucose to storing glucose, we send out a signal, a hormone. We send out the hunger hormone, which I can feel now. This hormone is called glucagon. It's produced in the alpha islet cells of the pancreas. It binds to the cell membrane. It activates an enzyme cascade. And this enzyme cascade uses ATP, chops off two of the P's, and produces a weird molecule of AMP, which has sort of cycle, where the phosphate actually binds not only to one of the OHs from the ribose, but to two. This molecule is called CAMP, or cyclic AMP. CAMP now binds to the R unit So CAMP binds to the R unit and the catalytic and the R unit dissociate. And while pKa was inactive with the R unit bound, now it is active without the R unit. So glucagon actually produces indirectly this CAMP. This is also called a second messenger. Second messenger, the first messenger is the glucose. Is the glucagon. First messenger is the glucagon, that is the signal. Actually, please release some glucose into the bloodstream. Don't use it for energy, because my brain cells, as you can tell, my brain cells are getting a little bit slower. They need the glucose. Give me some glucose, right? Otherwise, I'm in trouble. So glucagon is the primary first messenger. But instead of acting directly, it produces about 1,000 molecules of this CAMP. And this CAMP is again a messenger, an effector, but now we not only have one molecule, we have 1,000 molecules in the cell. So it acts, second messenger act as amplifiers. You could probably compare them to influencers on Instagram. So it's not just, one, if I post anything on Instagram, nobody takes any notice. But if I manage to get 5, 6 of the influencers to repost it, then the world will know. That has this amplification effect. So, CAMP is produced, it activates protein kinase A. Protein kinase A phosphorylates phosphofructokinase 2. The kinase activity of phosphofructose kinase 2 will be shut down. Instead, we have the phosphatase activity. So we go phosphatase. Everything ends up as fructose 6-phosphate. Fructose 6-phosphate is no longer an activator of PFK1. PFK1 does not become activated. Glycolysis does not happen. Whoa, that was a beautiful cascade. There are loads and loads of similar cascades and regulations, but we will discuss them when we get to the individual reactions. Any questions? I see you on Wednesday for round 2 of that.